Hey everyone, today we will be talking about the Barton decarboxylation reaction and the Barton-McCombie deoxygenation reaction. So we will first start off with the Barton decarboxylation reaction, which is basically the radical decarboxylation of an ester. And to get our starting material, we will start with a carboxylic acid. This is needed because you need to form radicals and you can't just use this as our starting material because it's not stable enough, which is why we'll need to transform it into a chlorine. Then our arrows will go like this, which breaks off the chlorine, and we form something called a Barton ester. And you can see that right here. To start this off, to get our radical, we will have our tributyl tin hydride, which is initiated by AIBN. And then our radical will go something like this. It forms our radical carboxylic acid and something known as Barton product. And this right here is a very important thing to note because it's a really strong bond and it's also what drives the reaction. But basically what happens after is that we lose the carbon dioxide, which is the decarboxylation of this reaction, and then we get a radical, but you can't just also leave it alone, which is why we need a reducing agent, which will also be our tributyl tin hydride. Our radical will react something like this, or do something like this, forming our product. It can also react with another tin radical, meaning that the cycle can repeat. And so the next reaction we will be talking about is the Barton-McCombie deoxygenation reaction. And we start off with also an alcohol, similar to the other one. We have some base here, and then this will combine with our alkoxide will combine with carbon dioxide, which will give us this, and then electrons will move something like this, giving us a thioester. Is a stable starting material that you can also see right here. We have the same initiation from AIBN and from the tributyl tin hydride, which means our radical will also be something similar like this. How do you know the difference? Because it has similar starting materials. It's the fact that the Barton-McCombie deoxygenation reaction will have no carbon dioxide to lose, while the Barton decarboxylation reaction does. And then we get left with our radical R, and similar to our previous one, we also get our product right after, and then the radical, well, to get our product, you would need to do this, and the cycle can also repeat when it reacts with another tin radical. And so let's do an example to see if you can differentiate between the Barton decarboxylation and the Barton-McCombie deoxygenation. And so you can see that the reagents are similar for both, like basically the same, but there's no carbon dioxide here that we can lose, which means that it's a Barton-McCombie deoxygenation reaction.